Wrong one. Okay, can somebody confirm you can hear me?
Yes, we can hear you.
All right. Let me show you this. Now, um, can somebody confirm? Is this the last slide we had discussed? Is this the last slide we discussed, or did we discuss further?
I have it as that is the last slide that we discussed.
Okay, thank you. That means we're a little bit behind schedule. All right, let's take a look at the schedule. So today is February 6th, and we are supposed to be starting chapter 5 today. Oh, wait a minute. Today is February 6th, and this is Tuesday. No, I take that back. I'm looking wrong. So we are supposed to be on chapter 4. We're on schedule. All right. Anything else I need to discuss? I need to discuss a little bit about the grading. Let me stop sharing my screen. I'll bring up the grade book. I've started grading one of the labs, but I didn't get very far. Maybe about a third of the students are graded. I think that's lab 4. I did get graded extra credit study sheet. I don't know if I did that this weekend or the previous weekend. And extra credit study 2 I did earlier. Extra credit study 4 I did this week. I've not done extra credit study 3. I thought I would jump ahead and give you some warnings. I don't remember if I gave anyone warnings or not, but do remember when you turn in your extra credit study that you should have everybody in your study group study for at least 40 minutes. Put your full names in, the date of when you studied, for how long you studied, and then the description of what you studied. It should be at least 2 sentences discussing what you're studying. You should not be studying your lab or working on your lab. Do that in your lab. You'll get credit for your turning in your lab. And remember, you should be asking each other questions. Some of you probably are doing that, but you didn't put in your extra credit study that you're asking questions. Do put that in your question, that you are asking each other questions. And it doesn't need to be the entire time. It just needs to be some of the time. It could be just a few questions. All right, I think that's it for that one. And let's take a look. I know I did not grade the lecture because the lab will take me more time. Extra credit quiz 2 is graded. I haven't gone in there to look and see if I'll give anyone extra credit on that. So lab 0, lab 01, lab 2, lab 3, and lab are graded for everyone for the first time. Nobody's graded a second time. And about a third to half the people are graded in lab 4. I'll try and get to the rest of the people in lab 4 today, so that lab 4 will be turned in for the second grading this Saturday. Lab 5, I seriously doubt I'm going to get it graded early this week. So I'm going to open it right now and say that the next grading, the until date, will not be this Saturday. It will be next Saturday. And that's assuming I get it graded sometime this week or early next week. So instead of February 10th for the second grading, that one's going to be pushed back to the 17th. I'm falling behind a little bit on the grading, and it's because I have so many students this term. Any question about any of that? All right, if not, let me get out of the grade book and I'll share my screen. There you go. Should be seeing my screen again. All right, so we talked about inclusions and stated that they are something that the cell is accumulating, and there's so much of that material in the cell that it actually precipitates out of solution, creating what looks like an inclusion. And it is a, I don't know, a precipitate of solution, meaning a solid in the cell. We need to talk about endospores. Endospores are a survival structure that some cells make when they are in a harsh environment and begin to die. Endospores are specialized resting cells produced mainly by Bacillus and Clostridium. There's at least one or two other families that also make it, but there's only like one or two species in these families. There's a lot of species in the family Bacillus and the family Clostridium. They are cells that have very low metabolism, shown here. And then one cell, the parent cell here, or let's blow that up. The parent cell here makes one endospore. The parent cell then, because it's in a harsh environment, will go on to die. One parent cell will make one endospore. One makes one, the parent dies. That's not reproduction. Any question about that? An endospore is a survival strategy, not a reproductive strategy. Endospores are something that can survive desiccation, heat, chemicals, radiation, and they survive conditions that the vegetative or parent cell cannot. The parent cell is metabolically active. It is reproducing, undergoing normal metabolic cell activity. The endospore is not. Like I said, it's a resting cell, and it's encased in a hard, I don't know, envelope or cell. What do you call that? Spore. We're just going to call it spore lining. And it can survive many, many things. Germination is when the endospore is in a favorable environment. It will germinate and turn into a vegetative cell, undergoing the normal metabolic activity, growing and reproducing of the parent cell. Sporulation or sporogenesis is the formation of the endospore. And that happens whenever the parent cell, the environment starts deteriorating and becomes detrimental to the parent cell survival. Let's watch a little bit of a video, not much, of endospore formation.
Grammarly's top 3 tips on starting an essay.
Let's say you're struggling to put pen.
All right, there's the endospore, obviously. Let me blow this up. An endospore forms much like cell division. The cell starts to divide, but it doesn't divide evenly. Splitting down the middle, it divides in a smaller portion than the larger portion. The larger portion, the DNA first replicates and moves to the opposite ends of the cell. And then the cell divides in a small portion. The plasma membrane will then surround what will become the endospore, and this will continue to be the parent cell here. The spore septum forms, which actually forms two membranes around the endospore. And you'll note that this portion here has now become part of the parent cell. So this moved in a little ways. The peptidoglycan layer then formed between the two membranes, forming sort of a cell wall. And then the spore coat forms over the endospore. And then the parent cell is in a detrimental environment and it'll die. And once that happens, it will release the endospore. Any questions about endospores? Remember, they're not a reproductive strategy. One cell produces one endospore, and then that parent cell goes on to die.
When you stain those, do those look dark like that picture, or are they more neon like the video showed us?
The video is showing you neon because it's showing, oh, what do you call that? Not neon, but like chromatography. It's a chromatography video where they're specially staining the cells to show the endospore. When you normally stain them, they'll look like this here, where the cell will pick up the dye. The endospore will not, because it's resisting incorporation of the endospore into the dye. We talked in the lab about the endospore stain. It will get in the endospore, and then you wash out the dye from the parent cell, and the parent cell will become clear. The endospore will retain the endospore stain, and then usually you stain the parent cell with the counter stain. Okay. All right, now we've come to the cell structures of eukaryotes. There are different cell structures in plant cells and animal cells. Most of the structures are the same. Yep, it's a little too big. Let me see if I can go back. Shrink that. Ah, that's way too small. Well, I'm just going to end the show and show it this way, and then can blow it up this way. Many of the structures are the same, like the mitochondria, the Golgi apparatus, the nucleus, found in both animal and plant cells. The chloroplast is only found in certain plant cells, meaning the green plant cells. The root cells do not have chloroplasts. If you're talking about the trunk of a tree, it will not have chloroplasts. Cell structures do include both membrane-bound structures, meaning a membrane-bound organelle like a perioxisome, the mitochondria, the Golgi complex, as well as structures that lack a membrane, such as the pericentriole and the centriole. I suppose the flagellum lacks a membrane. I'm not going to talk about the plant and animal cell structure, meaning the structure of eukaryotes, because you've already had that in your preliminary biology class. It would just be a review. It's here for you in case you need a review. One thing I do want to talk about is that animal cells can move and plant cells as well, by more than just the flagellum. If the prokaryote cell is swimming or moving, it is because that cell has a flagellum or several flagella. For eukaryote cells, they may move because of a flagella, but they may also move because of cilia, shorter hair-like structures where there's many of them on the cell. They usually beat in unison. Or they may move by pseudopods, which is not shown here, but the cell, remove the flagellum here, can move this way by putting out a pseudopod here, pulling up cytoplasm here, and then putting out more pseudopods, more cytoplasm here, pulling it up there. So eukaryotes may move by pseudopods, by flagellum, and by cilia. And prokaryotes may only swim by flagellum. Any question about any of that? I don't remember if the textbook has this correct or not, but there is a cytoskeleton. It's very extensive in eukaryote cells. It's a protein background running throughout the cell, making a backbone of the cell. And so here would be the cell here. You're not seeing it there, but it would be here. And there's the, actually, this is the cytoskeleton by F-actin. And what is this here? Microtubules in green, actin filaments in orange or red. And there's the cell there, and you're seeing the cytoskeleton here. There are 3 different cytoskeleton elements. You don't really need to know that, but it's very extensive, the cytoskeleton in eukaryotes. There is a cytoskeleton in prokaryotes. It was discovered about 20 years ago now. I'm not sure your textbook mentions that correctly. If they say there's no skeleton, cytoskeleton in prokaryotes, it's a mistake, and the author just hasn't updated it. But the cytoskeleton was discovered in prokaryotes about 20 years ago. It's not as extensive as in eukaryotes, but it is a framework of proteins running throughout the cell, giving the cell some shape and, I don't know, serving to hold the cell together and move things around too. I already talked about ribosomes. Let me briefly mention that eukaryotes have an endomembrane system, and that is where the ER, particularly the rough ER, but also the smooth ER, can put out vesicles, including things that are made in the ER. And then the vesicles can merge with the Golgi apparatus and then releasing the products made in the ER to the Golgi apparatus, where the products will be modified. You can methyl the groups, you can remove amino acids, things like that, and be sent through the Golgi apparatus, when then they will be transported by a budding off of the Golgi apparatus, which will be a vesicle, to move the contents to some location in the cell. And while the vesicle is moving, we call it a vesicle, an organelle that's actually transporting things from one location to another in the cell. But if the vesicle then just moves to a location and then stops and stores the contents in the cell, we call that a vacuole. And a vacuole is a vesicle that is just storing material. A vesicle is a vesicle that's actually moving material around in the cell. Any question about any of that? Altogether, this makes the endomembrane system, and that's the ER, both the rough ER and the smooth ER. The vesicles carrying the material to the Golgi apparatus. The Golgi apparatus modifying the contents in the vesicles, which are mostly proteins, but they can be fatty acids or lipids. And then the vesicles which bud off of the Golgi apparatus to transport the material to some location in the cell. And if it stores it, that vesicle then becomes a vacuole. So a vacuole and a vesicle look the same. They may even have the same. The only difference is function. The vesicle is moving the contents. The vacuole is storing the contents. All right, let's talk about the endosymbiotic theory. The endosymbiotic theory is a theory to explain the evolution of mitochondria and chloroplasts in eukaryotes. These organelles are different from the other organelles, largely in that they contain DNA and ribosomes. Other organelles do not have DNA or ribosomes inside of them. And then these organelles also have two membranes around them. For the chloroplasts and the mitochondria, we call the inner one the inner mitochondrial membrane, and then the outer one the outer mitochondrial membrane. And you can use those terms for chloroplasts. I think the inner chloroplast membrane is also called the thylakoids. And that term's a little more common in the chloroplasts than the inner chloroplast membrane. Well, the theory is that eukaryotes obtain the mitochondria in the chloroplast by engulfing a bacteria. And then that bacteria later evolved into the mitochondria, or another bacteria evolved in the chloroplast. And that's the endosymbiotic theory. Let me call up paint and then discuss this theory. Initially, we had a large proto-eukaryote cell. It wasn't a eukaryote yet. It was the ancestor of a eukaryote. And it could produce ATP by fermentation, taking glucose and making 2 ATP by fermentation. We'll talk about that if we haven't talked about fermentation yet. But this is what the early prokaryote could do. And then it engulfed a bacteria. Let me put the bacteria here. Oops, wrong thing. Bacteria here, we'll make it a rod type cell. And this bacteria could perform aerobic respiration. Making from one glucose about 38 ATP. Well, the proto-eukaryote engulfed the bacteria. So we're going to put out pseudopods. Oops, wrong one again. Oh, I still don't have that. Those pseudopods grew and surrounded the bacteria. And so the bacteria came inside the proto-eukaryote. Let me just erase the whole thing now, and I'll draw it inside the cell. I need a bigger eraser is what I need. And for some reason, the proto-eukaryote did not initially eat the bacteria inside it. So there's the bacteria, and there's the organelle membrane around the bacteria. So we're going to call this a phagosome. What do you call it? A membrane-bound vesicle that includes something that the cell consumed. And it didn't eat the bacteria right away. And some glucose from the proto-eukaryote moved into the phagosome and into the bacteria. And the bacteria took the glucose and made ATP. And for some reason, the bacteria released some ATP into the cytoplasm of the proto-eukaryote. We don't know how much was released, and probably the reason why the glucose moved from the proto-eukaryote to the bacteria, and then the ATP moved from the bacteria to the proto-eukaryote was just that life was early back then, and the molecules were just moving around and diffusing and all sorts of things, and life wasn't controlling it, and it was just a little sloppy, and that's what happened. Now, the proto-eukaryote said, wow, I lost one glucose, and all I can make are 2 ATP, and I got a lot of ATP. We don't know how much ATP the bacteria gave to the proto-eukaryote, but we know it was someplace between 2. I need to get my text again. Come on. Oh, I go here. There. It was someplace between 2 to 38. And it was probably more than 2, a lot more than 2, and probably a little less than 38. And let's just say an average would be, what would be the average of that? 36, so 18. So let's just say 18. And we don't know what it was. Someplace between 2 and 38. And the proto-eukaryote said, geez, I lost one glucose and I got a lot of ATP. I can only make 2 ATP from one glucose. And it must have been this bacteria that made the ATP for me. I'm going to keep that bacteria around and have him make ATP for me. I'll feed it glucose, but I'll get more ATP this way than I can make. And the two started living together in symbiosis, the proto-eukaryote and the bacteria. The bacteria actually liked this home inside the proto-eukaryote because the proto-eukaryote fed it glucose, and then it in turn fed the bacteria ATP. So the two lived in symbiosis, and now the theory is they've lived together for so long, we no longer find this bacteria free living outside of a eukaryote cell. You only find it living within the eukaryote cell. And initially, we're going to talk about this as the mitochondria, and then the proto-eukaryote now has the mitochondria in it. And so instead of calling it the proto-eukaryote, we'll now call it the eukaryote. And anyways, that's the theory. Is there any proof for this theory? First, you'll note that something that's engulfed within a cell will be have its own cell membrane, as well as the membrane of the organelle vesicle, so that this bacteria should be surrounded by two membranes. And when we look at the mitochondria, it has two membranes, an inner mitochondrial membrane and an outer mitochondrial membrane. That's weak evidence to support the theory. Let's go through some more of the weak evidence. Here we go. We can find some other evidence in support of the endosymbiotic theory, and that is we can find protists, simple eukaryote cells, which actually engulf a bacteria and then live in symbiosis with the bacteria. This bacteria is photosynthetic, and it gets a home inside the, this looks like a paramecium, but it's a eukaryotic cell. So the eukaryotic cell gives the bacteria a home. It feeds the bacteria, and then moves to light so that the bacteria can get light. And then the bacteria engages in photosynthesis and then shares its photosynthetic products, mainly sugar, with the eukaryote cell. So the two actually do live in symbiosis, and we can see a eukaryote cell engulf a bacteria, and then they live in symbiosis. In reality, there's even a few, oh, what are those called? There are a few protists, amoebas, that lack a mitochondria. And these amoebas will engulf a bacteria and then live in symbiosis with those bacteria for feeding the bacteria, and then the bacteria make ATP for the amoeba. So they're actually protists, which will engulf a bacteria and then use the bacteria like a mitochondria. The mitochondria, if you don't know, makes ATP. And the bacteria make ATP for the amoeba. So that's another piece of evidence for the theory. When we look at the DNA of a eukaryote cell, the DNA is linear. When we look at the DNA of a prokaryotic cell, especially a bacteria, there's generally one molecule. It's circular. And when we look at the DNA of a mitochondria and a chloroplast, there's one molecule and it is circular. If we look at the DNA of eukaryotes, the DNA is around histone proteins. When we look at the DNA of bacteria, there are no histone proteins in the bacteria. When we look at the DNA of the mitochondria and the chloroplast, there are no histones in their DNA. When we look at the first amino acid made in protein synthesis in eukaryotes, it's methionine. In bacteria, it's a different amino acid. It's similar, but it's different. It's formylmethionine. When we look at the first amino acid made in protein synthesis of a mitochondria and chloroplast, it is formylmethionine. When we look at the ribosomes of eukaryotes, it's 80S in size. When we look at the ribosomes of bacteria, they're 70S in size. When we look at the ribosomes in mitochondria and chloroplasts, they are 70S in size. When we look at the way that eukaryote cells grow, they grow by mitosis. Obviously, in reproduction, they grow by meiosis. Prokaryote cells, when they're growing, they grow by binary fission. The mitochondria and the chloroplasts grow by binary fission. So all of this is evidence stating that the mitochondria and chloroplasts are actually bacteria. And then the greatest piece of evidence for the endosymbiotic theory, when we sequence the DNA of eukaryote cells, the DNA falls within the domain eukaryote, which differs from the domains archaea and bacteria. When we sequence the domain of the bacteria cells, they always fall in the domain bacteria. So the sequence of these cells, their DNA is in the domain bacteria. When we sequence the DNA of mitochondria and chloroplasts, their DNA sequence falls within the domain bacteria. Very strong evidence that mitochondria and chloroplasts are bacteria. Any question about any of that? Now, what's interesting is for animal cells, this happened one time, and the bacteria evolved into the mitochondria. And now that bacteria is no longer found in the world outside of a eukaryote cell. It is always living in symbiosis with the eukaryote cell. In plant cells, that happened not once, but actually twice. One bacteria went into the plant cell and became the mitochondria, and that may have been a precursor proto-eukaryote that then split into plants and animals. That's probably what happened, but it could have happened that a plant cell engulfed a bacteria at the same time or similar time that a animal cell did, and then they both went on to evolve into the mitochondria. And then later, the plant cell engulfed another bacteria, which was a photosynthetic bacteria, and this bacteria evolved into the plant cell's chloroplast. Any question about that? Anyways, that's the endosymbiotic theory, and there is strong evidence to support that theory. All right, if there's no questions, that's it for chapter 4. Let's just begin chapter 5. I will promise that we will stop early and you'll get out of lecture a little early, but it's only 5:40 and we're supposed to go to 6:20. I want to go to close to 6. Guess we can close that down. Chapter 5, Microbial Metabolism. This slide is giving you the major goals on the rough outline. Know the terms. What's catabolism, anabolism, activation energy? What's an enzyme? An active site to an enzyme. What's competitive inhibition, non-competitive inhibition, also called allosteric inhibition? What's a redox reaction? What are photoautotrophs, photoheterotrophs, chemoautotrophs, and chemoheterotrophs? We'll talk about fermentation, cyclic and non-cyclic photosynthesis. Mainly in this lesson, we'll talk about aerobic respiration, what it is, what its steps are, and how it differs from anaerobic respiration, and then understand what feedback inhibition is. Any questions about what we're going to cover? All right. So microbial metabolism is all of the chemical reactions occurring within an organism. Now, when we're talking about metabolism of a eukaryote or eukaryotic cell, we say it's all of the chemical reactions within a cell. When we're talking about microbes, the cell is the organism. And so you could say all of the chemical reactions within the cell or all of the chemical reactions within the organism. Metabolism can be divided into two parts, catabolism and anabolism. Catabolism is a decomposition reaction where a larger molecule is decomposed into a smaller molecule, such as starch, a long, big molecule broken down into its monomers, like glucose, a much smaller molecule. Catabolism is energy releasing because a larger molecule contains more energy in the chemical bonds of the molecule than the smaller molecules. So catabolism releases energy. Some of that energy that is released as heat. Heat is just another form of energy. And as you might know from the second law of thermodynamics, whenever you change one form of energy to another, like the energy in starch to the energy in glucose, some of that energy is always lost as heat, and heat is just another form of energy. So heat is given off by the reaction. That reaction also gives off other energy, some of which may be used to phosphorylate ADP, meaning add a phosphate group to ADP to make ATP. Let me state that in animal cells, so in our cells, our body can only do this in two chemical reactions, aerobic respiration and fermentation. Animal cells are not very good at making ATP, and there's only two chemical reactions or processes that can make ATP, and that's aerobic respiration and fermentation. All of the other catabolic reactions going on in our cells do not make ATP. And we have a lot of catabolic reactions, such as, well, for example, when you take starch and you break it into a smaller starch molecule and you break off one glucose, release the glucose into the blood, that does not allow the cell to make ATP. What you've done is you made a larger starch molecule into a smaller starch molecule and one glucose. Okay? Actually breaking starch into all of the different glucose molecules, you cannot make ATP out of that either. The only way you can make glucose is when you aerobically respire glucose, breaking it down into CO2 and water to make ATP, or you can ferment glucose, breaking it down into pyruvate to make ATP. So there's only two reactions in animal cells to make ATP. All of the other catabolic reactions, and there's probably thousands or maybe even hundreds of thousands of them, do not make ATP. So this is a little oversimplified. An anabolic reaction is a biosynthesis reaction where you're taking a small molecule and you're combining them to make a larger molecule, such as when you take glucose and you make starch out of it. This is a energy using reaction. Energy must come into the reaction because the glucose molecules have less energy than the starch. So you need to add energy for this reaction to occur. Even though you're adding energy to make an anabolic reaction occur, and that energy usually comes from the breaking down of ATP to get the anabolic reaction to occur. Even though you're adding energy in there, you are changing energy from one form, glucose, to another form, starch. And so whenever you change energy from one form to another, some of that energy is going to be lost as heat. And so you're adding energy into the equation, but some energy is coming out as heat. Any question about that? All right. Metabolic pathway is when you simply add a sequence of a different chemical reaction and you add it to another chemical reaction, to another chemical reaction, to another chemical reaction, to another chemical reaction, making a metabolic pathway of several chemical reactions. In a cell, all metabolic pathways are mediated by enzymes. So the metabolic pathway is a sequence of enzymatically catalyzed chemical reactions in a cell. We have a starting substrate, molecule A. It'll be converted by enzyme 1 to molecule B, which will be converted by enzyme 2 to molecule C, which will be converted by enzyme 3 to molecule D, converted by enzyme 4 to make molecule E. Molecule E will be converted by enzyme 5 to make molecule F. Molecule F is the last product made, so we call it the end product of this metabolic reaction. Molecule A is the starting molecule, so we call that the starting substrate or the starting reactant. All of these molecules are called intermediate products. Molecule B is the product of enzyme 1, but it is the starting substrate of this metabolic reaction by enzyme 2. Any question about any of that? All right, so metabolic pathways are determined by enzymes. Enzymes are encoded by genes. So this all relates down to your genes. Any question about any of that? All right, this slide is showing you all of the metabolic pathways that we know of in a cell. And you only need to know all of them. Any questions? That's a little bit of a joke. You don't need to know all of them. Chemical.
I was going to say that's quite a lot to remember.
Yes. These are all the chemical reactions, though, that are going on in a cell. And you can see it's quite a lot. Cells are metabolically very complicated. Chemical reactions occur by the collision theory. The collision theory states that a chemical reaction may occur when atoms, ions, or molecules collide. The chemical reaction will proceed if there is enough energy in the collision to change the configuration of the starting molecules, atoms, or ions to make the end product. Activation energy is the energy needed to disrupt the electronic configurations in the atoms, ions, and molecules to make the end product. The reaction rate is the frequency of collisions with enough energy to bring about a reaction. So the reaction rate would be the frequency of the reaction, and that will be the collision that has enough energy to make the reaction proceed. You can increase the reaction rate by increasing the abundance of enzymes. That will bring the enzyme in closer proximity to the starting substrate, so the two will get together to make more products, and so more products will be made more quickly. You can also increase the reaction rate by increasing the temperature. When you increase the temperature, you increase the movement of the atoms, the ions, and the molecules. And if they're moving about more rapidly, they will be more likely to collide. And because they have more energy, when they collide, they will also be more likely to proceed or have enough energy to have the chemical reaction proceed. So that's how increasing the temperature will increase the reaction rate. Increasing the pressure is a little more complicated to discuss, but it's where you put more of the molecules closer together in a given volume. That would increase the pressure. And because there's more of them, they're more likely to collide. They are under pressure, so they'll have more energy when they collide, but you can increase the reaction rate by increasing the pressure. And that's sort of how a pressure cooker works. Not talking about the temperature, but when you're increasing the pressure, you increase the chance that the chemical reaction will occur with enough energy to proceed. So it speeds up the chemical reaction. And that's part of the way the pressure cooker works. Pressure cooker also is heating, so that would increase the chemical reaction as well. Any question about any of that? All right, here we're looking at a chemical reaction where the two reactants, which looks like it's one molecule, splits into two separate products. The reactant has more energy than the products A and B because the AB complex is a bigger molecule. So the AB products have less energy, and you can actually, on the y-axis here, this would be the level of energy. The products have less energy than the reactants. This chemical reaction will proceed, but you need to increase the energy of the reactant, getting it over an energy, what do you call that? An energy hump. And this we call the activation energy without an enzyme. The amount of energy the reactant has to have before it will break into the products and the chemical reaction will proceed. So that's a considerable energy hump. We can decrease that energy hump by adding an enzyme, in which case the activation energy is less. This we call the activation energy with an enzyme. It does need more energy from the reactants for the chemical reaction to proceed, but it's less energy than if the reaction were to proceed without the enzyme. One example of this would be to take glucose, a beaker of glucose here. We'll put it this way. Actually, this does have glucose in it, but and you can take the glucose and break it down into CO2 and water, the products. And this reaction actually does proceed because this has higher energy than that. And so every once in a while, glucose will break down into CO2 and water. But it doesn't happen very often. If I were to have this same container here tomorrow, it would be essentially the same glucose. So it would take hundreds of years, but eventually the glucose would break down into CO2 and water. And that's because what would happen is, what is that called? The particle from space, what is that called? Not an alpha particle, but a cosmic ray will hit this molecule, giving this molecule enough energy to get over the activation energy without the enzyme, breaking that molecule down. In this case, glucose, breaking it down into CO2 and water. And that does proceed, but it doesn't happen very often. If I were to look at this container tomorrow, you see the same amount of glucose and the same amount of glucose, even essentially a year later, there'd be very little decrease after even a year. But I could take a blowtorch to this, adding enough energy to convert all of the glucose into CO2 and water. And that's obviously the amount of energy you need to add, and that's the temperature of a blowtorch. But yet glucose, all the time in the room you're in, is breaking down into CO2 and water. Where is that happening? Where is glucose breaking down into CO2 and water all the time in the room you're in? Come on, somebody's got to guess.
Inside of our bodies.
Yeah, inside your cells. Glucose is breaking down into CO2 and water all the time. Are your cells at the temperature of a Blowtorch, that's around 1,000 degrees Fahrenheit, by the way. It might be 1,000 degrees Celsius. I don't remember. 1,000 degrees, something like that. Awfully hot. Obviously, your cells are not that hot. They're only 37 degrees Celsius. And that tells you that the reaction rate, the activation energy with an enzyme, is considerably lower than the activation energy without an enzyme. Because glucose is breaking down in your cells all the time into CO2 and water. Any questions about that? All right. I think I'll stop here. That's a good place to stop. And we are actually, I believe, yeah, we're a little bit ahead of schedule, so I'm going to stop here. Any questions? All right, if there's no questions, I will see you in the lab for those of you who are coming to the lab. And the rest of you, I'll see you next Thursday.
Thank you.
All right,
Thank you.
bye.
Thank you.
Bye.